Dear students, I am Dr.V.Violet Dhayabaran, Head of the Department of Chemistry, Bishop Weber College. I would like to introduce the concept sensors, which comes under the topic electrochemistry. Sensors are widely used in day-to-day life as we are all driven by technology advancements from Industry 4.0 revolution. Lot of devices have come into practice. Without sensor, we could not make use of all the facilities in the environment. So it is very necessary to understand the principle of sensors, the types of sensors, the field at which the sensors are used for different purposes. So that is why I have prepared this presentation, which includes the basic principle, types, and as well as the essential features of sensors. In addition, the specific examples given for electrochemical sensor and biosensor. I hope this presentation will be very useful for your real-time applications. Sensor is nothing but it's a device that detects and responds to some type of input coming from the physical environment. So based on this principle, lot of devices have come and in various departments like transportation and logistics and industrial applications and in agriculture sectors and animal tracking, environmental monitoring, energy control system, food safety, and in security and surveillance, healthcare and monitoring. In all the departments, we have been using lot of devices which involves the principle of sensor, and there are so many smart sensors have come in real time applications. As I already discussed, sensor is a device that detects and responds. The input will be coming from the environment and that will be converted into electrical signal, and this electrical signal will be given as output, which will be readable form. So it involves a processing that will be taken by the inbuilt system present in sensor. Actually, this kind of smart molecules will be designed based on the structure of the molecules. So those molecules will be having advanced structure and they must be able to detect the stimuli from the physical environment with high sensitivity and high selectivity. So these are the important parameters to be understood by every researcher before making a smart sensor. So smart sensor are smart molecules that can easily detect the stimuli with high selectivity and high sensitivity. In such molecules, there are a lot of different kinds of properties that they possess should be examined. Usually, these molecules will be synthesized, tailor synthesized based on the applications of the society. In such case, the magnetic properties, optical properties, catalytic properties, electrical properties, all those things will be examined thoroughly. Then only they can be made or designed as smart sensor. This is a diagram showing how this kind of molecules are working. When we supply some input from the physical environment as chemical quantity, that will be taken by the sensor molecule, and when it takes that input, a transducer will be there in the sensor and that will convert the analytical signal. This kind of mechanism is working in sensor. There are different types of sensors are available: chemical sensor, electrochemical sensor, optical sensor, biosensor, and colorimetric sensors. I would like to highlight the devices that are used in different types of sensors. When you consider optical sensors, the photo detector, pyrometer, infrared sensor, proximity sensor, these things are working based on the light intensity received by the sensor. Therefore, this optical sensor will measure the light intensity based on the type of input that will be converted into electrical signal, and the result or the final output will be given as useful information, which will be used to calculate lot of useful information in the industry field. This is a type of sensor called water quality sensor. Nothing but a sensor which can measure the pH, turbidity and conductivity of water. Not only in water but also the type of foodstuff that we are using in industry or in the market, that can be tested using this kind of sensor. In health sectors, healthcare sensors are used, especially brainwave sensor, even the smart watch that we are using in the day-to-day life, able to detect what is our heartbeat and how many steps we have taken. All those things will be taken based on this type of biosensors. Even we can also include nanoparticle. So when we integrate the nanoparticle in the synthesis or designing of sensor, the quality of the sensor will be enhanced, because this nanoparticles are having very high novel property, and that will increase the sensitivity and specificity. Therefore, in biosensors, nanoparticles are included. Therefore, nano biosensor are widely used and it is very popular in the society. If you consider any molecule, the quality, the standard of the substance, or you can say sensor, will be evaluated based on these features. So there are certain parameters are fixed to evaluate the quality of the sensor. For example, number one is sensitivity. Sensitivity is nothing but how this particular sensor can even sense very low concentration of substance. So how good it is, that can be evaluated based on its sensing ability. The next one is selectivity. If you have a mixture of different kinds of substances, and how far this sensor is able to detect a specific substance. So in the midst of different kinds of substances, our sensor molecule must be smart enough to identify certain specific substance. So that can be evaluated. That is called selectivity. And response time, how it is quick, speed of detection, how far it works very well, the signal generation, that will be evaluated. That is called response time. The next one is detection at nanomolar concentration. The limit of detection that will be evaluated. And reproducibility. After performing an experiment, when we repeat it again, we must be able to get the same type of results. And easy handling. It should not be very complicated or complex. Therefore, if you want to evaluate the quality and standard of a sensor, these are the parameters will be used to evaluate it. Sensitivity, selectivity, response time, detection, reproducibility and easy handling. These are the different kinds of pictures shown to highlight how these parameters can be measured. To measure the efficiency, we have to plot a graph. For example, in this first graph, we can see the response time or sensitivity with respect to temperature. At what particular temperature, the sensitivity is very high. that can be identified. Suppose if you prepare a group of substances with a different percentage of certain specific substance, we can evaluate which particular substance will have high sensitivity by plotting the graph like sensitivity versus temperature. If it is a sensor used for the industry. In the same way, if you go for any biosensor, we would like to estimate what would be the minimum concentration at which the sensitivity will be very high. For that, using cyclic voltammetry, I versus E graph is drawn. Here it shows different peaks based on the concentration limit, starting from 0.5 micromolar to 100 micromolar concentration. From the height of the peak, it is easy for us to identify which particular concentration is very good to fix the sensitivity. So, these are the different kinds of measures we take, and these are the graphs showing the selectivity in the midst of different kinds of substance, how my sensor will be smart enough to detect a specific substance. Here, the sensor molecule is made up of a hybrid composite molecule, that is manganese dioxide, a polymer polyaniline and graphene oxide. So this particular composite is used to modify the glassy carbon electrode, and it is tested for finding the sensitivity of the sensor to detect methyl parathion, which is a pesticide. Here, in this case, a wide range of concentration is given, that is around 20 nanomolar concentration. In this graph, we can see in presence of different metal ions, calcium, zinc and nickel, how far this particular sensor is able to detect the target molecule, that is methyl parathion. We can see the peaks at A, B, G, H. It shows specific concentration of methyl parathion present in the system. Like that, we must evaluate how far this particular sensor is effective and efficient based on this kind of testing. So, selectivity, sensitivity, these are the two important parameters to be studied. When we consider the sensor, we have to understand the components present in the sensor. There are two things, one is called actuator, another one is called transducer. There are a lot of differences between the sensor and the actuator. We always focus on sensor molecules because as chemists, we will be interested to select the molecule and design a sensor for a specific purpose. In such case, how this actuator will be useful to design a specific sensor. Sensor will only detect the changes in the environment, and it will convert that change into another form of energy. But in actuator, a component of machine that will be attached to it, and that is responsible for moving and controlling mechanism. This sensor will be connected to the input, whereas the actuator will be connected to the output. The sensor will be active to absorb the changes in the environment, but the actuator will be very helpful to control the output system. In sensor, electrical signal will be produced based on the changes in the environment. But here the output movement will be controlled by actuator. So in the case of biosensor or chemical sensor, the sensors are completely used along with the actuator. So it is nothing but, actuator is nothing but an electric motor. This is the schematic diagram showing how the components present in the sensor are working. So from the measuring quantity, the changes that occur in the physical environment will be fed as input. The sensor will detect that and it will respond and it will convert into electrical signal. That will be, whole thing will be taken care of by transducer using a circuit system controlled by computer system. And after that, the transducer will convert this electrical signal into another form and we will get meaningful information as output. So transducer that will be taking care of conversion of energy from one form to another form. So there are a lot of signals, circuits are present that will be inbuilt in the sensor and the transducer is attached with sensor. So without transducer, the sensor will not work. So the role of transducer is very important in the functioning of sensor. So this is the working principle of sensor highlighted. Analyte is fed into the sensor. Based on the interaction between the analyte and the sensor, you will be getting some signal that will be captured by the transducer, converting that form of energy into another form. And this will be amplified, recorded. And that information will be given as a data and it will be recorded in the output. So when we consider the different kinds of stimuli from the physical environment, it may be either resistance change or capacitance change or voltage generation or light modulation, whatever may be the change occurred in the environment, that will be taken as input and that will be detected by the sensor, then should be processed. After the processing, we will get the information. So this is the basic principle. When we consider biosensor, the scenario will be slightly different. The analyte will be a biological component. So biosensor, we will be having biological components as input and the sensor molecule will be very specific to this biological component and that will sense based on the specificity and that form of energy, combining energy will be converted to electrical signal, again that will be converted into readable form as output. When we consider the biosensor, the important points to be remembered is, number one, it is an analytical device, converts a biological response into electrical signal, detects, records and transmits it to another physiological change. And whatever change converted, that will be converted into electrical signal and finally we will identify what would be the useful information that we arrive from this biological system. So this is the working principle behind biosensor. When we consider the biosensor, there are two important types. One is called catalytic biosensor, in which enzymes, microorganisms and tissue samples are used. Whereas in affinity biosensor, antibodies, nucleic acids, DNA, RNA and hormones will be used. So these are the very important biological components acting as sensor based on the usage and application purpose. This kind of substance can be selected to design the biosensor molecule. Actually, I am happy to introduce the father of biosensor named as Professor Leland Clark. Even in the period of 1918 to 2005, he has formulated this kind of device which will be very useful for identifying different kinds of diseases. And based on the selectivity and specificity of the biological substances, how it recognizes the substances, the information will be detected and that will be recorded. Therefore, biomolecules can act as sensor, either enzymes or microorganisms or DNA or RNA or any hormone which are all present in our body can act as a biosensor. So in the medical field, lot of biosensors are used to evaluate the diseases and to identify the problems to solve the physiological problems. And when we consider the components of biosensor, there will be receptor. The sensors are used as receptors, the other name given for biosensor. So either it may be enzyme, antibody or microorganism, and the transducer that will convert these changes into electrical signal. So semiconductors can be used as transducer or electrodes can be used. All those things are inbuilt in sensor, and we are enjoying the application of this biosensor by having the good result from the detector. So this basic principle, number one, first biological recognition will be taken place. Second, it will convert the transducer from one form of energy to another form based on the biological target. And the third one, it will amplify the result and give useful information in the display board. So these are the steps involved in biosensor. When we consider a biosensor, how the quality or the standard can be evaluated. So the efficiency of any biosensor can be evaluated based on its linearity. That means when we increase the concentration of the analyte, then there should be increase in the sensitivity. So sensor ability should be very high when we increase the concentration. So the detection limit will be linearly proportional. And another one is sensitivity. So the electrode response per substrate concentration. So that is very, very important. Not only the biomolecule, but also the electrode which is responsible to detect. That is very, very important. And selectivity. So based on the nature of biological component, we have to select certain molecules. So chemical interferences must be minimized so that selectivity would be very high. And the response time, how quick it is. The speed of the sensor is also taken into consideration to measure the efficiency. I would like to highlight the biosensor which is commonly used, very often used in our day-to-day life. So if you want to check the glucose level, suppose if any diabetic patient is in our house, we would like to record the amount of glucose or sugar level at different times periodically. So in such case, how this biomolecule works as sensor and how it is detected, how we are able to relate the glucose content in the body and the reading that we read in the monitor. When we consider the glucose biosensor, we will be given a strips, and these strips will have a coating of an enzyme that is nothing but glucose oxidase. So if we want to find out the sugar level of any patient, we will prick and we will get the blood, and that blood will be allowed to fall on the strips in which the glucose oxidase substance will be coated. What would happen? In the blood, the glucose present in the blood that will react with this enzyme, and immediately it will be converted into gluconic acid, and it involves 2 electron 2 proton transfer. So when this is converted into gluconic acid, based on the number of electron involved in the reaction will be measured in terms of the glucose concentration in the blood. When the glucose content is very high, the amount of gluconic acid produced will also be very high, based on which the number of electron transfer and the amount of gluconic acid produced will be increasing. That will be detected by the electrode and it will be converted into electrical signal. So, the amount of glucose present in the blood is directly proportional to the amount of gluconic acid produced as well as the number of electron transfer. When it is converted into electrical signal, then we can see the reading very well on the display unit. So, this is the mechanism working in glucometer, or you can say glucose biosensor. I would like to highlight a case study. We have formulated a substance, designed a substance, and it is a composite material made up of manganese dioxide, a polymer, and graphite. Why we have mixed these three? All the three substances are giving synergistic effect to enhance the sensing ability. So this particular substance is prepared and it is coated on the electrode, glassy carbon electrode, and it is used to detect methyl parathion. I hope you are well aware of this methyl parathion used as a pesticide to control pests in the fields. It may be agriculture field, vegetable or fruits, whatever may be. Because of this, harmful effect may come if it is not washed properly. In order to maintain or ensure the quality of food and free from all the harmful substances, chemical substances, we have introduced this particular sensor to detect methyl parathion. This is the research work that we have done in our laboratory. So, based on the nature of the substance, how far this detection is taking place. So, it is used as an electrochemical sensor. As we all know, why this kind of detection is required? Because the pesticide that I have quoted, methyl parathion, is responsible for different kinds of harmful effects like allergic sensitization, oxidative damage, asthma, gastrointestinal disorders, neurological disorders, Alzheimer's disease, Parkinson's disease, birth defect, genetic disorder, reproductive disorder, DNA damage, kidney failure, cancer, or for all the problems are associated with the organophosphorus pesticide methyl parathion. So, it is very important to formulate or develop a sensor which can detect even very low concentration pesticide methyl parathion present in the food system. Already lot of research work is going on. So, different kinds of a group of pesticides like organophosphate, organochlorine phosphate, carbamate, these are all pesticides used in regular practice, and these are the toxic effects given by different pesticides, and what are the different kinds of electrode materials are used, and what are the methodology adopted to detect these kind of harmful substances. So, this research work is also published in journals. Development of highly selective and sensitive electrochemical sensor based on nano hybrid. So, it is also nanoparticle, manganese oxide, polypyrrole and graphene oxide, reduced graphene oxide, and it is used for the detection of methyl parathion. So, while we do this particular sensing, we have used cyclic voltammetric study, that is a electroactive nanocomposite will be pasted on it, and we followed different kinds of instruments, experiments, and these are the different kinds of polymers have been used to identify which is very effective. So, to examine the electrical properties, we carried out electrochemical impedance study. To examine electrochemical behavior, we carried out cyclic voltammetry study. And to quantify the detection of methylparathion, we carried out differential pulse voltammetry. And to assess the stability and sensitivity, we used amperometric analysis. So based on different kinds of study, we have to evaluate the efficiency of the sensor. So this has been published, and even it is used to evaluate the amount of methylparathion present in fruits and vegetables. And these are the graphs that we have drawn to identify the selectivity, sensitivity, and as well as the response time. And this graph showing in presence of different kinds of metal ion, zinc, nickel, sodium and calcium, how far this particular sensor is capable of detecting even very low concentration methylparathion, and how the sensitivity increases with respect to concentration shown here. And we carried out everything using cyclic voltammetric method. Therefore, I versus E graph is shown for different concentration. See, this is the table showing for cabbage and grapes, strawberry, all those things, real life applications. We took all these different kinds of substances and we added selective amount of substance of methylparathion, and we evaluated what is the sensitivity and the detection limit and what is the linear concentration range. All those things have been evaluated. We found that it is very effective, even very nanomolar concentration, 10 power minus 9 nanomolar concentration of methylparathion can be detected using this particular sensor. My dear students, hope you have understood what is meant by sensor or smart sensor, how to design it, what are the important parameters to assess the quality, what are the different types of sensors, how it can be used, and what is meant by electrochemical sensor or biosensor, the principle behind these sensors and how it can be effectively used, and the examples also we have seen. And the specific case we have seen the glucometer and as well as the polymeric nanocomposite are used to modify the electrode. When we consider the quality, standard applications of sensor, not only advantage but also we have little disadvantage. So this slide shows smart sensor have both pros and cons. So it is very easy for us to detect very nanomolar concentration, easy to design, and we can use and maintain and monitor, very reliable information we can get it, the performance is very good, minimum interconnecting cables, everything is okay. So all are positive. There are also certain difficulties we have because it contains both actuators, sensors, very complex circuit, and the cost wise it is little higher, and calibration, it requires calibration before using it, and is always embedded, inbuilt in the substance. Therefore, there are little difficulties, but when we consider the advantages, it is more than the limitations. Therefore, it is well used, very well used in the society, and in every sector we have been using the sensor effectively. As we are all in chemists, it is in our hands to develop very high quality sensor, smart sensor for different purposes. Hope this presentation will be useful, design new molecules and for different purposes. So the points that we highlighted: smart sensor, types of sensor, working principle, parameters, and real world application, case studies, pros and cons. This would be highly useful for your future study when you go for material chemistry or electrochemistry, how we can make use of the instruments to assess the quality and how we can propose or design a new molecule for future studies. Thank you very much.